This video is on learning target 9, meiosis. This is the second, or beginning of the second section of unit 7 on genetics. To understand meiosis, we first have to get an understanding of chromosomes and kind of how we label cells and talk about cells. In prokaryotic cells, remember, there's only one chromosome. So when we talk about meiosis, we're not only talking about eukaryotic chromosomes because there's more than one chromosome, but we're also talking about eukaryotic chromosomes because with meiosis assumes organisms are sexually reproducing. This is only occurring in sexually reproducing organisms. Prokaryotes are not. And so we are assuming that we're talking about sexually reproducing organisms wherein that organism has two parents. Most cells in a eukaryote are 2N or diploid, which means that they have more than one copy of chromosome, meaning that they get one copy from mom, one copy from dad. In humans, we have 46 chromosomes in our average cells, in our normal cells. We call these cells our somatic cells or body cells. Somatic cells. Well, there are 46 chromosomes or 23 pairs, one from mom, one from dad. Well, in our sex cells or our gametes or sex cells, these are called haploid because they only have a single copy of the chromosomes, and this is what we pass on in either our sperm cell or egg cell that combines with another cell, and that makes a full diploid zygote like we've talked about before. And so you have to understand when we're talking about meiosis, we are talking about the formation of these haploid cells. Meiosis is the formation of haploid gametes. So here's a picture of that. Meiosis, we start with a cell that is 2N, so one diploid cell. And what we're going to end up with is 4 haploid cells. And these cells, and you see this right here in this picture, these cells are all genetically different from one another. All right, we'll talk about how that works. So you start with one diploid cell, you end up with 4 genetically different haploid cells. This is completely different than mitosis, which we'll talk about those differences in a moment. Another big thing I want you to notice about this picture, when we talked about the cell cycle and mitosis came at the end of the cell cycle, and it began for the 2 new cells that were made, began a new life cycle for them. Well, in meiosis, we have 4 cells that are made, and those cells are the end result. Those 4 cells, whether they're sperm cells or egg cell, those cells will not do anything else. They will just, that's the end of the process. They then wait for reproduction. Now in meiosis, there are 2 divisions. So in meiosis, there are 2 divisions. And we're going to start meiosis with interphase, which is not one of those divisions, so don't mean to confuse you here. During interphase, however, the DNA is going to be replicated, so it's very important. What I mean by replicated, I don't mean cut in half, I mean doubled. It's going to make a copy of itself, replicated. All right, the first division is called meiosis 1, and the second division is called meiosis 2. Pretty easy to remember those, meiosis 1 and meiosis 2. Now the phases for these are going to be similar to ones that we've seen before. Of course, interphase, again, not part of meiosis 1 per se. I don't really like this terminology, interphase 1, even though it's widely used, because there's no interphase 2, and we'll talk about that shortly. But the phases of meiosis 1 then are prophase 1, metaphase 1, anaphase 1 and telophase 1. Very similar to mitosis, but there's going to be obviously some big differences. And so I'm going to draw this. So here's our cell. In this particular cell, we're going to have one homologous pair of chromosomes. We get the red one from mom, we get the blue one from dad, we'll say. This is a homologous pair, meaning that they are very similar to one another. They are homologous to one another. They have the same genes, but they are still two copies of that. All right. We have 23 pairs in our cells. This cell has one pair. Well, interphase is going to occur. And during interphase, those homologous chromosomes are going to replicate. So this is when we get this shape that we're familiar with, this X-looking shape. Now, here are still two chromosomes, but each of those chromosomes has two sister chromatids. The two red pieces, the two blue pieces, each one completely identical to one another. The blue identical to the blue, the red identical to the red. And so we have essentially doubled the amount of DNA in this cell. You could say, if you wanted to say it this way, if this was a human cell with a diploid, here's the diploid cell. I mean, you could look at it whereas this becomes 92, because 46 times 2. And I know I just said there's only two chromosomes there. However, there are 4 copies of that DNA, and so there's not in a human cell there would be 92 copies of the DNA. Now I'll use these 46 and 92, and when you divide those again, there's 23, and I'll use those numbers a lot. But to understand that not all organisms have that many chromosomes. But we're going to use those because we're going to talk about human genome quite a bit. And so the next thing that's going to happen is the first division. Remember I said the first division is called meiosis 1. Well, there are several things that have to happen in meiosis 1. And the first thing that happens in meiosis 1 is called prophase 1. Now, during prophase 1, 2 very important things are going to occur. Well, very similar to prophase and mitosis, whereas the chromosomes are going to form, the DNA condenses to form the chromosomes, the nuclear envelope disappears, and so you don't have a nucleus anymore. However, different things. These 2 homologous pairs are actually going to kind of line up very close to one another so that similar parts of each are touching one another, kind of like lining your hands up together so that your fingers line up perfectly with each other. This structure here that is formed is called a tetrad. The prefix tetra means 4, and so here we have 4 pieces of DNA that are lining up together, this tetrad. All right, and when tetrads form, you can have a phenomena called crossing over. And so let's go over here and just show you. So make this a little bigger to show you an example. So this is that tetrad. Well, they are so close to one another that what ends up happening is the pieces of DNA, because they're similar, they're homologous to one another, they will actually swap. And so what you end up looking at after prophase 1 is you'll have the red chromosome that looks like this. So I had a knock on the door. Sorry about that. And so what ends up happening is the red will actually take part of the blue, and the blue will actually take part of the red. So you have some mix up like that. You have some crossing over is what that's called. And that only happens during prophase 1 of meiosis 1. So during metaphase 1, they're going to line up in the middle. During anaphase 1, this is another very important part because this is something that's often confused. During anaphase 1, we're not quite 2 cells yet, but I'm going to just go ahead and make it 2 cells. During anaphase 1, these homologous pairs are going to separate. You don't have the blue separating from the blue and the red from the red. You have the blue separating from the red. You have the homologous pairs separating, so that one cell is going to get dad's DNA, the other cell is going to get mom's DNA. All right. And so then telophase one, you get the cleavage furrow forms, cytokinesis happens, and viola, you end up with two cells. And that's meiosis one. So in meiosis two, a very similar kind of thing. Well, let me make one more note here that's very important actually. Because up here we started with a diploid cell. And here at this point, when it was back where I had it, that was diploid too. But now, do we have diploid or haploid? We have haploid cells because this cell only has half of the original complement of DNA, as does this. Now they have a copy of that half, but it's still only half. And so those are haploid cells. Now during meiosis 2, the big thing, prophase 2, nothing really going on there that of note. The nucleus disappears and all that's going on again, but there's no crossing over during prophase 2. There's no tetrads forming because we don't have any tetrads. We don't have enough DNA to have tetrads. And so that's not going to be happening. Anaphase 2, however, there's something that you need to note that is different than anaphase 1. In anaphase 2, the sister chromatids are going to separate. And so telophase 2 then, and cytokinesis, what you end up with are 4 cells. And if you account for crossing over and some of the other things that we're going to talk about with recessive and dominant alleles and that kind of thing, you have 4 genetically different haploid cells. Now, in males, this is what produces sperm. So in males, you get the production of 4 sperm cells here. Pretty simple. You have interphase, meiosis 1, meiosis 2, 4 sperm cells. One diploid cell makes 4 sperm cells. However, in females, it is different. In females, what happens is this first division, with the first division, you have the production of a tiny little cell called a polar body. And this cell actually ends up getting most of the cytoplasm. All right. Well, then you have further divisions. You have these little polar bodies. And with that second division, with meiosis 2, you have another polar body that is formed. So the polar bodies here, here, here. And then what you have left is the big cell called the egg. Now, what is a polar body? These polar bodies are just tiny little cells. They're haploid cells. They have DNA in them, but they're not being used by the body. The body just dissolves them back and takes back the parts. And this is so that the egg can have the majority of the cytoplasm, so that the egg can be a giant cell, because when the egg combines with the sperm is not bringing any cytoplasm with it. And so it needs that extra cytoplasm for the first several cells that are going to be made in the new embryo. So again, sperm production, one diploid cell makes 4 haploid sperm cells. In egg production, one diploid cell creates 1 egg cell and 3 polar bodies that are going to be dissolved back into the body. Now, last thing, differences between mitosis and meiosis, just to go over these again. How is it different? In mitosis, 1 cell, 2 genetically identical cells. This is where you have the daughter cells that are being created. In meiosis, you have 1 cell that makes 4 genetically different cells, haploid cells at that. And in mitosis is part of a cycle. Mitosis represents the end of that cycle in which those cells will begin that cycle again. In meiosis, you have the end, you have a process that ends with a product, the product being the haploid gametes or the haploid sex cells.